I'm Tebb. I'm delighted to be back here again. I joined EMBL in November 1985, and it was interesting because I think the member states had pressured EMBL to start a program to train young scientists. So the year before I joined, there was the first intake of PhD students. I believe there were 6 of them. They were obviously fairly good, and the next year EMBL decided to take in another 6, and I was one of them. We had a fairly entertaining selection procedure, and then in April, I think, and then we started in September, October.
In those days, EMBL wasn't as well known as it is today. What persuaded you to come to do your PhD at EMBL?
The main reason I came to EMBL was actually because I wanted to come to Germany to learn German, to understand Wagnerian opera better. So I applied for 4 PhD positions throughout Germany. I think I was accepted by all of them, funnily enough. I still don't understand why. And EMBL was the lab I visited that made the most favourable impression on me.
And what was your PhD project?
I was working in Ian Mattei's group, and in those days Ian worked both on transcription and on function of US and RNP particles, and I was the transcription side for a long time. So the transcription of small nuclear RNA genes.
And what are your memories of that time at EMBL? It was a time in the gene expression program when we had a large Italian contingent working.
We were separated in the gene expression program into a small outbuilding called the containment. Originally it was contained, gene regulation studies were contained there, and no genetic engineering could be brought into the main building. It was completely separate. Riccardo Cortesi, as you remember, was the head. Sadly passed away a few years ago and sorely missed by many of us. And it was essentially a little Italian outstation. And I think when you joined the containment in those days, you bought into the life. You played football, Italy against the rest of the world on Sunday morning. You would have dinner with the Italians in the evening. Someone would disappear into the kitchen, produce a plate of pasta. Someone else would run off into the lab to take samples for everyone else or put things on ice. It was a mixture of social life and science life, and there was really little life outside EMBL or outside the containment.
The community at EMBL at the time was much smaller. The physical infrastructure was much smaller, but as a PhD student here, how do you feel it prepared you for the rest of your career?
Well, the first thing I should say is that the small size has a huge advantage, which is that because there were so few PhD students, we were fairly tightly networked. We had weekly, bi-weekly meetings, seminar series. Everyone knew everyone else, and everyone knew all the experiments that were going on in their labs. So as a result, even as a PhD student, I had the feeling that I could talk to pretty much anyone at EMBL. I knew everyone. I could talk to anyone about their science, and they could talk to me about my science. And I found this input from very different disciplines extremely stimulating and very helpful. And I think that's what helped me very much when I went on to work as a postdoc.
So your career post EMBL has moved in a slightly different direction from bench research into the administration and funding of research. Could you tell us a little bit about what you do today and how your experience at EMBL maybe has informed your thoughts about how science is managed?
Possibly the single most important thing I learned about EMBL is that quality is key, and quality will come out. So I believe that all universities, all research institutes should aim to do high quality research, high quality basic research. The fundamental aim is to advance knowledge. It's not up to us really to decide how that knowledge should be applied. Our goal is simply to tackle problems that we find interesting and to come up with innovative and unexpected solutions. I think EMBL has taught me the importance of quality, and in my present job I'm supposed to be helping scientists at my university, the University of Veterinary Medicine in Vienna, design projects and acquire third-party funding to carry them out. I think my focus on quality from my time at EMBL has remained, and I'm always encouraging people to not to do the next logical step, but to tackle the interesting problems and tackle the questions that can really make a difference.
Yes, I completely agree with you. I think I'm very passionate about the importance of defending basic science and to put the main emphasis on excellence in whichever endeavour that you carry out, because we simply don't understand today what it is we need to know to be able to solve the problems of the future, but there's increasingly pressure to perform mission-driven projects that are perhaps easier to sell to governments or funders. Have you seen that playing out in Austria? Have you seen it playing out in other European countries in your experience? And how do you feel about the tension between doing projects with a much more defined mission and explaining the importance of doing, high quality basic research?
I fully agree with you that both aspects of science are important. You need to tackle immediate problems, but you need to allow scientists scope to tackle what they think is interesting and what might become important. The problem with tackling immediate problems is that scientists are by their very nature creative, and any good scientist will be able to explain how what he or she is doing is immediately relevant to the question of interest. So a lot of this mission-directed research, in my opinion, is merely repackaging of what was going on anyway. There are clear cases. COVID was one wonderful example where people needed to solve the problems of how this virus enters cells and replicates and goes on, and people needed to develop vaccines for it quickly. So I can understand in crisis situations diverting a lot of basic research funding into particular areas, but I think we need to be very careful that we don't define too many crises and allow scientists the freedom they need to do what they think is interesting and challenging.
Yes, I completely agree with that. Is that a message that it's possible to sell to our political masters who fund our research?
The trouble with our political masters is that most of them are on very short timescales. Their primary concern is whether they'll be elected in 3, 4 or 5 years. And if you tell someone that you're working on the basis for an advance that might occur in 20 or 30 years' time, their concern is that some other government will derive the credit for that. So it is a very hard message to sell. Ultimately, it's a question of trust, and all we can do is point to how the work we did, our predecessors did in the 50s, 60s and 70s, has laid the groundwork for the enormous progress that we're enjoying today.
What advice would you give to a young scientist if you think back to the young Graham Tebb arriving at EMBL, fresh-faced, enthusiastic, in a very different environment? You've now had the experience of training, working in different areas in different countries. What advice would you give to a young scientist today starting out as to how they should go about building a successful career?
I think when we're thinking about advising people, we need to remember that careers are far less linear than when we were young. Sorry to put it like that, but we are no longer young. We may think of ourselves as that. People have much less linear career structures, and I would encourage people to do what they find most interesting and most challenging at the time, not to worry too much about where that will lead, but to trust to fate, if you like, or in your own ability. And I think clever people will generally do good things and find sensible pathways. So I would advise people not to allow themselves to be pressured too much into doing what other people think is right for them. They should follow their own instincts and believe in themselves.
I think that's a great answer, Graham, one I completely agree with. In my own case, I had the benefit of ignorance. When I started off, I was sufficiently ignorant of career structures and the low probability of being successful that I never worried about it. I just kept doing things that I was enjoying at the time. And I think that's good advice to give. So putting yourself back in that position, you are the young Graham Tebb, and you've come back from the future to advise you. What project or what field of science do you think the young Graham Tebb might like to research if he was starting out today?
I often wonder whether the young Graham Tebb would be proud of the old Graham Tebb and what he's accomplished in the meantime. And
I'm sure he would.
Thank you. It's hard to say. The young Graham Tebb was fairly demanding and fairly critical, and the older Graham Tebb has become a lot more able to see advantages of alternative approaches. I think the most important thing is to be true to yourself and to do what you really believe is right. It's important what people think of you, but it's more important still what you think of yourself. We have to bear in mind that possibly 0.5% to 1% of postdocs who graduate and and appear fresh-eyed and keen on the job market. Only about 0.5 to 1% of them will end up becoming full professors or group leaders at EMBL. And the danger is that 0.5 to 1% are the people who will be telling everyone else what to do. So it's important when you're starting out on a career to look at options outside science or outside pure research and think whether you might find a place there where you can contribute as much and feel happier and more settled. So strive for excellence in everything, consider a career in academic research, but be open to other options.
Yeah, I think that's excellent advice. I think the training that you get prepares you for a wide range of other options, and we definitely shouldn't be encouraging people to think that an academic career is the only one they could pursue. And a great thing with the interactions with alumni is to see how many successful people have built fantastic careers using the skill sets they've developed in many other areas, including science communication, teaching, and all sorts of other areas. So I think that's really important. Is there a particular area of science at the moment that you follow closely or that you're especially interested in beyond transcription of snow RNA genes?
You've asked me this before and I've managed to dodge it once. Because of my work as a research manager at a university, I have to follow all the areas in which my university is actively researching. We are a university of veterinary medicine, but we're not confined to veterinary medicine, so we do basic biology, we do human-animal interactions, we do population genetics, we do an awful lot of cancer biology, a lot of signals transduction. I have to remain vaguely up to date with all of those areas so I can discuss research plans with the scientists who want to do the research, and that's both challenging, there's an awful lot to read, and very stimulating, of course, because I have the advantage of being semi-involved in a lot of areas without having to do the experiments myself, which is, if you like, one of the things that frustrated me most about science. My memory is that I wasn't completely hopeless in the lab, but even so, it was frustrating when I knew the result of an experiment and I had to repeat it 3 or 4 times simply to have prettier pictures so that they'd be better for publication. So I wasn't learning anything, I was merely doing cosmetic changes. In my present position, I'm exposed to the pretty results. The people do their experiments lots of times, come and show me their data. So I have the thrill, the stimulus of looking at the data without all those months of agony of having to generate them.
Yes, well, that is certainly an inevitable fact of real-world bench science, that not everything works and you sometimes have to repeat things. I think we're coming to the end of our time slot. I would just like to finish up by giving you a chance to leave us with any particular abiding memory of the EMBL. When you think back to your days when we were both young in body as well as in spirit, and we were having a lot of fun here, what is it that you most clearly remember from those days?
I remember, and he will love me for saying this, in an Italy against the rest of the world football match, Roberto Di Lauro scoring one of the best goals against me of all time. I still maintain it was a fluke. He curled it with his instep and it hit the edge of the post and went in, and I will never forget his celebration after that goal. I remember Paolo and Alfredo walking past, wheeling a trolley and then managing to appear a minute later from the same direction but in a different order. I remember your seminar talk, A Tale Told by an Idiot, your favourite Macbeth quotation, under which one of the Italians had written 2 idiots. I could go on at length. I remember that it was a very, very intense period. We worked, I believe, really hard. I think we were successful. If I look at the publications we produced in the 3 years I was here and compare them with my publication output later or with the publication output of PhD students now, we were phenomenally successful. But my abiding memory of EMBL was that everyone had a lot of fun all the time and science was really enjoyable and really stimulating. We lived and we breathed science and we loved it.